Well, Matthew gave a very elegant description of the output signal. I will give hopefully an equally elegant view of the input signal. The disciplines that are sort of required for understanding touch, you can see here from a paper that Galadi and Spence published a few years ago. You can see how multidisciplinary this area is of touch. As a neuroscientist, I'm just obsessed with mechanisms. I always want to know how something works, and then that helps me interpret the behavior in a more predictive way. So I will be giving you some understanding of the basic processes that are the input signals to Matthew's output signals. And the bottom line of my talk is here. The recently discovered system of unmyelinated nerves in the skin regulates stress and the immune system. Here are the key things: lowers heart rate and cortisol levels, releases endogenous opiates and oxytocin, and so much more. Now, this is a pretty tall order for one nerve fiber, but hopefully I'll convince you that that is what it does. A couple of sort of basic sort of lessons in terms of the different peripheral nerve fibers that innervate the skin. Basically, we have fast ones and slow ones. I seem to have dropped the slide here. So the fast nerves are myelinated, and the myelinated nerve basically sends information to the brain in a matter of milliseconds. So if a fly landed on your nose, you detect it immediately. The other major population of nerve fibers in the skin are C fibers, and they're unmyelinated. An unmyelinated signal can take thousands of milliseconds, if not a couple of seconds, to get into the brain. So in terms of any defense mechanism, the fast myelinated nerve fibers clearly do a good job. But what are these C fibers doing, the slow ones? Well, that's what I'm going to unpack a bit. A bit of basic anatomy from this rather complicated slide. We have two types of skin. Glabrous skin is basically the palms of your hands and the soles of your feet are glabrous skin, and there are no hairs on those particular body parts. The rest of your body is covered in hairs. Even though you may not see them, these very fine down hairs, they're all over the body. And as you can see on this image on the left, you can see how these hair follicles has the most beautifully exquisite innervation. They are innervated by a whole population of different classes of nerve fibers, the myelinated ones, the unmyelinated ones, and in particular, a passion of mine for the last 20 years is a particular unmyelinated nerve fiber that basically provides the reward associated with physical contact. So this innervation, this complex innervation, isn't there for no reason. So why is the hair follicle so beautifully innervated with all these different populations of sensory nerve fibers? Just as I just mentioned, the idea of these fast and slow nerves is going to be quite critical to the way I understand. I explained a system which is predicated on basically driving the affective or emotional inputs to the brain, and that's what these C fibers do. A couple of examples, by the way, I always like to look at sort of comparative physiology. It just also brings in some other interesting insights into the mechanisms that are underpinning affective touch. So this is an experiment done by some Californian biologists. I suppose they had maybe they were trying to hide their MDMA, so they threw it in the tank with the octopus. And what this octopus did, the octopus is normally quite a solitary animal. So rather than sort of playing with its toys, the ecstasy or the MDMA in this tank changed these octopuses into spending more time touching each other. This is 500 million years of evolution separates the octopus from the human. But here we see affective touch regulating behavior in an octopus. If we step up the scale a little bit more to rat pups, this is a study which is looking at the impact of a maternal touching, licking and grooming behavior on a rat's subsequent development as an adult. And this study looked at rat pup mothers who licked and groomed their pups a great deal compared to those that basically ignored them. And those rat pups that got a lot of licking and grooming, again, stimulating this nerve fiber that responds to pleasant touch, grow up to be really chilled and not anxious rats, whereas the low touch group basically were stressed adults. The only difference is touch. That also affects the neuronal complexity in the brain as well. All of this stuff is published. We move on to Harry Harlow's infamous experiments where a monkey removed from the mother at birth, given the opportunity to be either sort of feed from a wire surrogate or cling to basically a useless cloth surrogate. And of course, the monkey spent most of the time, clinging to the cloth surrogate. There was no reward associated with that, ostensibly, But Harlan knew nothing about this nerve fiber I'm unpacking today. But again, this is pleasant touch. What's the mechanism? And I'm sure Robin will mention this in his talk later on when you look at grooming behavior in monkeys. And here we bring in another neurotransmitter. This is a study that Barry Coburn did many years ago. He gave one group of these grooming monkeys naltrexone, or an opiate blocker. The other group had a very low dose of opiate, and he looked at grooming behavior. Those monkeys that had the opiate blocker increased the amount of grooming. So we're getting a link here now that opiates are driven in some way to provide the reward associated with close physical contact and grooming behavior. And Robin's been involved in a pet study which showed a similar effect in humans. So all these are examples here, and there are many more. The loneliness one I haven't spoken about, but lonely people don't get touched. What's the mechanism? Again, we've got two types of nerve fibers. The first one we know a reasonable amount about already. So it maps onto the somatosensory cortex, where there's a map of your body in that part of cortex. And Matthew described the other side of that sulcus, which is where all the motor systems are. So the sensory and the motor systems are housed in this area. However, these C fibers they play a role in emotional regulation. And that's where they play this phenomenal role in protection. So C fibers are unmyelinated, slowly conducting nerves. And the two that we know most about are the pain, or the nociceptor, and the prioreceptor. These are C fibers that code for a nociceptive stimulus leading to the experience of pain, or the pruritic or itch stimulation driving the motor behavior of a scratch. However, these amazing Swedes are the people I owe my life's obsession to. They are the most incredible neurophysiologists. In 1939, Zoterman, recording from the saphenous nerve in the cat, identified a C fiber that was not a nociceptor. It responded specifically to stroking touch. There was a massive gap until another Swede developed this phenomenal technique of microneurography that I'll speak about in a moment, where they identified exactly that kind of C fiber in the humans. Only in the late 1980s and 1990s was this nerve fiber discovered. These are the C low threshold mechanoreceptors, the CLTMRs, and are hypothesized to code for the rewarding properties of touch. And this is just an image of Zoterman recognizing that when he stroked the leg of the cat, he saw this C fiber input coming along in seconds after he'd actually delivered the physical stimulus. So these C fibers are an evolutionary ancient class of nerve fibers innervating the skin of the body, the C afferents, and their primary role is one of protection. Now, they've traditionally been studied as separate entities, the itch nerve, the pain nerve, etc. But we're now seeking a synthesis of how these C fibers actually play together. And I'll briefly describe that when I'm talking about pain later on. And if you look at the anatomy again, the large proportion of peripheral nerve fibers, afferent peripheral nerve fibers, those that are sending information into the brain, are in fact C fibers. So the anatomy tells you something about it. There's just a couple of images here about how these C fibers play a fundamental role in protection. And this is an example of a damage to the skin, say an itch, a pain, something like that. And you see all these factors kick off. These nerve fibers are playing a fundamental role in sensing that signal, but they're also affecting a whole defensive mechanism where the blood vessels become dilated, there's a blister, basically that toxin is moved away. So that's the protective side. If we now look at the pleasure side, we have here, and this is this discovery of this new nerve fiber, new in terms of historical terms, where it responds specifically to gentle touch. So the fast nerves fire off, tell the brain that something's happened on the skin, and then you get these C fiber slow nerve fibers basically feeding this information. That's the fast stuff. And here's the slow C fiber. So that information gets into the brain maybe thousands of milliseconds after that gentle hug. So how do we discover? Well, we have the three techniques here that we've used in my lab to discover, and the Swedish guys, in terms of how we unpack what these C tactile afferents are all about. We've got microneurography, we got psychophysics, and we also use imaging. So these techniques allow us to unpack the complexities of these nerve fibers and basically what they do. Now, this robot that strokes touch that we've used in a series of experiments to tell us how these nerve fibers respond specifically to different velocities of touch. Now, this is the very first experiment that I think that was probably ever done asking any human being whether touch had any positive and emotional properties to it. And here the participant is being stroked at different velocities across the skin. They have a visual analog scale that they're rating how pleasant or unpleasant that stroking stimulus is. And guess what? When that stroking stimulus is stroked across the skin at around 5 centimeters per second, people will rate that as far more pleasant than a slower or a faster stimulus. And again, of interest here is the topography of this response. If that same stimulus is stroked across the face, they will rate that stimulus as far more pleasant, even though it's exactly the same physical properties. And this is a logarithmic scale. So stroking across the face, again at 5 centimeters per second, is rated as far more pleasant than stroking faster or slower. Now, here's the microneurography. So basically, we can wiretap into a peripheral nerve. So this is a peripheral nerve. These are fascicles, and within those fascicles are individual axons. And these axons connect to all the different receptors on the skin that you saw my previous slide. So the touch ones, the pain ones, the itch ones, the temperature ones. Using this technique, Okavalwa was able to wiretap in humans a C-tactile afferent. And this is the stimulation system. This is me lying there quite comfortably. These two electrodes here are going through my skin into the underlying nerve and listening in to various signals coming up that nerve. And that's how we find the tactile, responsive, slow touch nerve fibers. So we stroke across the receptor field of the unit, which is on this part of the skin, and you can see the signals that are feeding into massive motor cortex. And this is the input signals in response to pleasant touch. And that's a beautiful sight. Yeah, that's touch at the level of a neuron firing and it firing a signal. So rather than go too much detail, this is all published in a Nature Neuroscience paper a few years ago, where we spent 3 or 4 years recording from CTs, stroking across the skin. Here you can see the 6 different velocities of stroking. Here's the nerve's response, and we find that that nerve has a tuned characteristic preference to a stroking stimulus around 3 centimeters per second. Myelinated nerve fibers basically just encode linearly. The faster you stroke, the more they fire. We then do the psychophysics and ask people how pleasant was that, and we get exactly the same function. They rate 3 as far more pleasant than fast or slow, and that's the basis of our pleasant touch hypothesis. So we're pretty confident that we've discovered a nerve fiber here which is responding to the positive characteristics of a physical stimulus. This is one slide that we submitted a paper to, which I think will be very germane to discussions today. This is done in my lab in Liverpool and primarily in our Linköping group. The Swedes have been recording from these C-tactile afferents, and they're looking at the response of that nerve fiber to a hair that it is innervating. So every single hair we've ever recorded from is innervated by one of these C-tactile afferents. So when you deflect the hair, the blue line, you can see the nerve fire. When you brush across, it's also velocity tuned. And you can see how now, so this is an indication that these hair follicles are innervated by these reward processing C-tactile afferents. This again, I think is probably very relevant to the discussions today. And this is hair pulling. So we're recording from the nerve fiber with microneurography, and the hair is now being tugged out. And you can see how active that nerve fiber is beyond the actual pulling of the nerve fiber. That nerve keeps on firing. Now, if this is releasing all these endogenous opiates, etc., maybe we have a mechanism here which describes the reward associated with that stimulus. Getting into the brain now, this is a patient that many of us have worked with who's lost all his fast nerve fibers. So Ian has no sense of touch in terms of touching on his body, but he has C fibers. And when you stroke across his hairy skin, we see activation in paralimbic areas of the brain. So this is the first evidence that these C-tactile afferents are being processed not in the primary somatosensory cortex area, the discriminative brain. They're going into the limbic system, the affective brain. This is the wiring diagram, which I won't go into now, but this is all in the Neuron paper. So what is it about this pleasant touch system? So this is affective touch, slow conducting unmonitored nerve fibers found exclusively in the hairy skin. The optimal stimulus is gentle stroking. They're also beautifully temperature coded as well. So they will respond more to a skin temperature touch than a warmer or a cooler stimulus. They also are more active in terms of releasing oxytocin. So I'm bringing in some other hormones here. So 5 minutes of stroking activates the hypothalamic oxytocin neurons. We're seeing now the benefit of self-soothing behaviors in terms of the release of oxytocin through touch. This one is also relevant in terms of the response now in another brain processing area, the nucleus accumbens. In response to stroking, we get the release of dopamine. So there's a whole cocktail of really sort of pleasure-seeking drugs being released by these by this stroking touch. So just a couple of things now. So looking at the studies we've done, looking at the role of this nerve fiber in stress control. This is a study I did with a colleague of mine in Valparaiso. Alexei Danino had a very sophisticated rodent model of stress, testing drug anxiolytics, etc. I convinced Alexei just to take a population of rats every day and stroke them at 30 centimeters per second. Remember, this nerve fiber won't respond to that. Another group of rats were lifted from their cage and stroked at 5 centimeters per second. And there was a couple of control groups. For 2 weeks, for 5 minutes a day, you got the fast touch and slow touch rats. They were subjected to a chronic, unpredictable, mild stress paradigm. So a hectic 2 weeks, the smell of cats, light going on all over the place. The only intervention here is fast touch or slow touch. At the end of that 2 weeks, there were 2 separate experiments. We measured corticosterone levels. Then if you look at corticosterone levels in the rat pups that got stroked at 5 centimeters per second, they were down at the same level as a group of rats who were never stressed at all. The fast touch stroke rats, basically their corticosterone levels are through the roof. And if you look at their behavior, they were behaving in the slow stroke rats basically were stress resilient. And just one example of the elevated maze, you can see the rats that got stroked at 5 centimeters per second were resilient to any kind of stress associated with this particular test. 5 minutes to 10 minutes a day of stroking touch had built resilience in these rats. The pain one is another indication of how these C fibers basically interact. This is a study I did with Rebecca Slater at Oxford. I suggested she had developed a technique of measuring event-related potentials from term babies' cortex in terms of a heel lance, so a nociceptive stimulus. So I convinced Rebecca to basically stroke some of the legs of these babies at the preferred CT velocity touch. And what we found is that those that got that gentle touch before the heel lance, the evoked response, the nociceptive response evoked from the scalp has basically shrunk. So stroking has basically gated pain. And again, this may be relevant to some of the discussions that we're unpacking in terms of mechanisms that drive you know what I mean. So 2 sides of touch, discriminative effective, 2 systems. I now call this system vitamin T because it's absolutely a fundamental part of our daily lives. And boy, didn't COVID prove that to us. These are some of the people that I've been so fortunate enough to work with over the years that have helped me along this journey. And there are 3 major societies for these C fibers. The pain one you all know about, I'm sure. The itch one set up a few years ago, and then now we have a affective touch, which we should talk about, in fact, with Claire. And we published a book if you want to know more about it. And here's a whole load of animals having a really nice time being